I'd like to thank you for the invitation to give this lecture. I'm Dr. Gesvaldo. I'm speaking from Brazil. I'm professor of the Department of Biological Science of the State University of Southern West of Bahia, Brazil. And today we'll talk about the interaction between different pathways associated with insulin receptor, as you can see here, KTP channel, and peptide precursor amyloid. That is one of the most important peptide associated with the Alzheimer's conditions. But today we'll try to make a link between the insulin receptor and APP, that is a peptide, Alzheimer's disease, and autism disorder. I'm Gisvaldo Hugo Hawkins from the University of Colombia, Marcos Alves from the Colombia too, Erica Pereira and Djalma Menezes, colleagues of mine here at the university in Brazil. So the first question to address Alzheimer's, before to address my presentation, is to understand why to study Alzheimer's conditions. That is a good question. So Alzheimer's disease is one of the most prevalent disease in the world. It has a high elevated cost with care costs for the government. For example, just in 2018, in the United States, the government it spent something about almost $300 billion with care costs with Alzheimer's conditions. About in 2050, the care costs with Alzheimer's disease will step over to something about $1.1 trillion. This is just in the United States. It's a huge amount of money. And even after 100 years, 1 century, more than 1 century, since the early Alzheimer has diagnosed Agosto with Alzheimer's disease, almost nothing have changing because we don't have cure to Alzheimer. It has been changing to develop of vaccinations and the use of insulin and another approach, but in all the cases we don't have successful. But back into the Alzheimer, it's necessary to understand what is the main characteristic of the Alzheimer's disease. So in Alzheimer's disease, we have a dementia that has been characterized by the cognitive impairment, the presence of amyloid plates, the amyloid cascade that it may be resulted from the genetic mutations in the genes that will be responsible for the cutoff in the processment of amyloid peptide in surface of the cell. So I will show for you in animation from the NIH, the brain will use extensively the APP, the peptide that is not in the amyloid form. So we have the synapses between neurons, the synapses between different cells. Is necessary the presence of the amyloid peptide. We need to have its presence to bind one cell to another, as you can see here. So in a normal cell, it will be responsible for the cell growth, for example, for the memory formation and the perfect metabolism of the cells. I will accelerate just to get Alzheimer's disease. Okay, here I have the surface of the cell. It's very huge peptide in surface of the cell is the APP, the peptide. It's APP, it will act in the cell like a vacuum between both cells, okay? And it will tend to stabilize the connection, the synapses between the cells. So here I have the presence, the action of the alpha secretase and the gamma secretase. So this is necessary throughout the day to happen because its processment of the APP will be responsible for the perfect connection between the cells. Okay, so we have the normal process, and here is a normal fragment of A beta. But here we have just something about 18 residues of amino acids that will be different from the beta amyloid. So this is in a normal conditions. In, a, in Alzheimer's disease, we have a different situation. As you can see here, beta secretase will cut off the APP, and gamma secretase will release a fragment with 14 residues of amino acid. And during the density, it tends to form amyloid plates, senile plates, as you can see here. So this is one of the most features, characteristics of the Alzheimer's disease. But now we know that the presence of senile plates is not the cause of the Alzheimer's symptoms. We know that it's a consequence of the Alzheimer's symptoms. What was discovered recently is that individuals with autism conditions, we have the high level of the APP in the alpha form. I, I will back to show for you here. In individuals with Alzheimer's, we will have the presence of amyloid peptide. In individuals with autism disorder, we will have the presence of the alpha peptide, that is in blue peptide here, as you can see here. Okay, so for who don't know perfectly what means autism disorder, what is the characteristic, the features of autism disorder, we have the cognitive impairment in some situations, and autism disorder, or ASD, as we know, can be considered a neurodevelopmental disorder, of course, but it shares a common pathway, a metabolic pathway with Alzheimer's disease. And why is it important to understand its situation in a neurodevelopmental disorder with a neurodegenerative disorder like Alzheimer's? It's because in autism disorder, we have a macrocephaly. Macrocephaly is very different in Alzheimer's condition that we tend to have a disaggregation and the loss of brain mass. So our aim is to understand how the APP, the peptide, will, what is the role of the peptide in individuals with autism disorder, to try to transport its treatment to the Alzheimer's condition. Okay, so if I have individuals in autism disorder with a macrocephaly, a neural rigidity, so if I understand and try to make a reverse engineering, it may be possible to get a treatment to Alzheimer's disease. This is our aim in such a study. And why to use to study autism? Because in individual with autism disorder, it has been detected a high level of SAPP. SAPP is a huge fragment here that is used by the cell to stabilize the connections between the synapses and to prevent the loss of neurons. So our aim is to understand such process, to transport its process to the treatment to Alzheimer's disease, because we have exactly the opposite situations. Okay. Okay. So to address is the main characteristics and what is the pathway, metabolic pathway that will be shared by the Alzheimer's disease and individual with autism conditions, we have the presence of the full mood network. The full mood network is a complex that will be formed by a pool of areas inside of the brain. For example, the temporal lobe, the part of the medial prefrontal cortex, posterior cingulate cortex, medial and inferolateral cortex, precuneus, parietal cortex, and hippocampus. As you can see, we have a pool of areas, several areas inside of the brain. This is interesting because this pool of areas will give to the brain the possibility, for example, to work in a background. For example, when you are in a beach, just looking ahead the skyline into the sea, your the full mood network, it may be paradoxical, but it will tend to increase the consumption of glucose. And it's very interesting to investigate this because we know today that nowadays that 20 years before the development of the first symptoms of individuals with Alzheimer's, the consumption of glucose, it has been changing in the full mode network, in that areas of the full mode network. It's very interesting. But in another side, we have individuals with autism disorder that is exactly the opposite situation. Individuals with autism disorder, it tends to live most part of your time, of him time, in high performance of the full mode network, in high level of activation of the full mode network. This is one of the responsible for one of the most characteristics, the feature of the individual with autism disorder, that is the introspection. Because the full mode network that is used for us to catch the attention of our brain for some situation, individuals with autism disorder, we have a high level of activation of the full mode network. Okay, so to evaluate the situation, we decided to investigate its different pathway related to insulin signaling pathway and the SAPP or APP and beta amyloid, just to propose a treatment for Alzheimer's disease. So as you can see here, we have the peptide amyloid. I have a neuron here. This is a synapse. I have pre-terminals. Here I have the post-terminals, and we have the action of the APP peptide into the synapse. As you can see, it will take function like a Velcro that will tend to stabilize the connection between two different cells. So individuals with autism disorder, we have a high level of its connections. It tends to lead to impairments in certain functions because it will tend to reduce the neuroplasticity. That is exactly the opposite we have in individuals with Alzheimer's disease. And this is what we are trying to understand to apply in the treatment to Alzheimer's. Okay, so we have an investigation using is a theoretical approach. We have first, we decided to investigate the main genes associated with the metabolism of the APP and the insulin. We have used a neurogenomic approach. Neurogenomic, we use techniques from the mathematics, from the physics, from the computational studies, just to understand how different genes will be wired together in the same pathway. Okay, so first we will use Allen Brain Atlas, that we have a microarray of the different genes, and to understand how the different proteins will be bind between together in the same pathway, we'll try using a docking molecular approach. In the molecular docking, we'll try to understand how geometrically way how different proteins will be bind one to another. Okay, let's go. So here, I don't know if it's possible to see the resolution. I think it's not so perfect. but I think it's possible to see the insulin receptor, the APP, as one can see here. The APP is a name, the hub genes is the most connected genes to different metabolic pathways. In our situation, we were interested in the insulin pathway and the APP pathway. Here we have the different genes that will be associated with the same metabolic pathway. So this is the pathway that will be shared between Alzheimer's with APP, as one can see here at the middle of the figure, and here with the insulin receptor, as one can see here. So when we get the hub genes, now we will try to understand how that genes will be wired. Here I have an approach in a high resolution, and here I'm using a metabolic pathway associated with a metal. In this case, that was related with copper. Okay, I will see why copper and why metal. I will explain in the next slide. So here we can see in this obtained from the expression of the genes, of different genes. I have the APP expression inside of the brain. I have the ADAM10 that is responsible for the metabolism of the metals inside of the brain. And I have here the kinase protein. What is interesting to see here, to observe here, is that a different expression, different level of expressions at the same regions will be indicative that its different genes will share the same pathway at the same time, because this is obtained from immunohistochemistry in different parts of the brain. What is interesting is observe that the APP peptide will be associated with the cell growth in all the situations. Insulin into the brain will not take part as responsible for the metabolism of the glucose inside of the brain. Insulin will be main associated with the mTOR peptide, that will be, its complex will be responsible for the cell growth inside of the brain and for the progressions of connections between synapses, different synapses, and neuronal plasticity. In other words, we need insulin inside of the brain to promote the formation of connections between different memories, to perform different memories. So what we have discovered in our approach, in our theoretical approach, is that when you see here at the left, we can observe the beta amyloid peptide, and at the right we have the alpha peptide. Okay, what we see in blue is the pocket site where insulin will be bind in the receptor. This is in blue is the receptor of insulin. What is remarkable is that the beta peptide will tend to bind, as you can see here, exactly at the same site where the insulin will be bound to. And what that means? It means that individual with Alzheimer's, the peptide, the beta amyloid, will block exactly the receptor, the site where the receptor of where insulin will be bind. And what that means? It means that the individual with Alzheimer's condition, such situation, it will tend to decrease the formation of connections between cells, because in such situation, the insulin will not have possibility to execute its function, that will be to promote the cell growth to to increase the formation of memory or LTP or long-term potentiation inside the brain. In other words, the peptide, the beta amyloid, it tends to block the bind of insulin inside the brain. These are interesting questions. And as you can see in the right, we have the alpha peptide. We have a L1, these are L2, L1 site where the insulin will be bind. As we can see here, the alpha peptide, in a different way from the beta amyloid, it tends to act as a insulin in a progress, like a sum. It's like if I have a sum between different effects, different proteins. For example, insulin and alpha peptide will formation of connections. Now we are trying to understand how it's possible to use the alpha peptide to increase the stabilization of the synapses inside the brain of individuals with Alzheimer's disease. This is very interesting and one of the most remarkable results that we get, because if we can understand, if it's possible to understand how the alpha peptide bind to the insulin receptor and increasing the action of the insulin inside of the brain, it's possible, maybe it's possible to prevent the loss of cell connections that we have in individuals with Alzheimer's conditions, as we can see here at the left side. Okay, let's go ahead. Here we have the amyloid here, beta amyloid, and at the right, it's possible to observe the site where some amino acids will be bind to the peptide. So what we have with a theoretical approach is the surgical approximation of the sites, because it's possible to understand how and what who the different amino acids will take part into the bind of the in this case amyloid beta peptide and the pocket site of the insulin. So here we have the alpha peptide. As you can see, is very different from this. Can you see how the beta amyloid block completely the pocket site where the insulin will be bind? So seeing in alpha peptide is completely different. So it tends to increase the action of the insulin. So the full mode network, what we can depend from this is that from the full mode network, we have the mechanism that will tend to act as a neuronal protection. What that means? It means that if you try to understand how the full mode network may be interactions with the insulin receptors or complex mTOR and APP, it's possible to derivate its relationship of the insulin and APP to increase treatment for Alzheimer's disease. Okay, let's go. It's not interesting to see. Okay, I told before that the presence of copper inside the brain that is necessary as oxidative stress, to promote oxidative stress in different parts of our body. In our brain, we have the most huge amount of copper in our body. As you can see here at the figures, this is a microscopy, electronic microscopy of the brain and the formation of APP amyloid aggregation. In A, we have the aggregation, the formation of senile plates. And when we don't have the presence of copper, as you can see, we tend to have a high amount of formation of senile plates. That is exactly what we will see in individuals with Alzheimer's. Okay, this is another address that we are trying to understand how to use the metabolism of metals like a copper to improve the treatment of individuals with Alzheimer's. And where the relationship with individuals with ASD, with individuals with autism disorder, is that individuals with autism disorder tend to have impossibility to metabolize copper and iron and other metals. In other words, individuals with autism disorder tend to have a high concentration of copper inside the brain. But the amyloid peptide have exactly its peptide here, the alpha peptide, okay? It has exactly a copper site here in this position. In other words, the APP peptide, it acts like a binding, a bind of copper and metals inside the brain. In individuals with autism disorder, we have difficulty to metabolize copper and iron. But in another word, they show a high amount, a high concentration of APP peptide here. What that means? That means that individuals with autism disorder may be using or overexpression APP peptide, alpha APP peptide, like a counter reaction to stabilize or to reduce the high level of metal inside your brain. Because if we have a high concentration of metal like copper, it will tend to formation plates, as you can see here in figure A. So in another situation, we are trying to understand how copper will be, what is the role of the copper inside the brain of individual with Alzheimer's, individual with and individual with autism disorder, to try to transport the treatment from the individual with autism to individual with Alzheimer's disease. It's like a puzzle, okay? So our findings suggest that the high concentration of SAPP or of SAPP amyloid will be a consequence of the oxidative stress generated by abnormal level of copper inside the brain of individual with autism disorder. In other words, the downregulation of insulin receptor related by the casein kinase may be one another consequence of the presence of the high levels of the beta amyloid. So the high level of SAPP, as you can see here, it will tend to increase the stabilization of the synapse. So we are proposing the use of high levels of SAPP alpha in individuals with Alzheimer's disease, just to try to stabilize and to increase and to preserve the formation of synapses and to stabilize the old synapses and to increase the metabolic pathway between insulin and mTOR that will tend to increase the formation of new connections. This is our aim, and that was what we found in our theoretical experiment. Okay, so that is, thank you so much